Swayam Prabha. Digital India. Educated India.
Hello everyone. So today we will start conducting system of heart. So in this topic we will study what are the subtopics we have. We will cover different parts of the conducting system of the heart, the AV nodal delay, explain various mechanisms of the AV nodal delay, the conduction velocity in different cardiac tissues, and how the electrical impulse is propagated along the conducting system of heart. Now, generally the muscles of heart consist of two types of myocardium, the normal myocardium and the modified myocardium. The normal myocardium, which is striated in nature and consists of actin and myosin filaments, specifically myosin 2. This myocardium is mainly responsible for the contractile properties of the heart when the heart contracts. But when we talk about modified myocardium, this is mainly responsible for the conduction, that means conducting the electrical impulse from one part to the other. So, we have basic differences between these two types of myocardial cells. Now, normal myocardial, that is atrial and the ventricular myocytes, they are usually singly nucleated, they have one nucleus. They are connected each other with intercalated disc. Now, we had already studied in the previous lectures that each cardiac myocytes, the two cardiac myocytes are usually connected electrically as well as mechanically. So, electrical connection is through the gap junctions, that is electrical window, and this gap junctions are nothing but they are the electrical synapse which is made up of connexin proteins. Also, we have the mechanical connections through desmosomes, and this help the cardiac cells to contract as a whole, and hence this is known as the functional syncytium. Now, if we talk about the modified myocardial system, which plays a very important role in conduction, it has got less actin and myosin filaments, because the main role is not contraction, conduction. So, it should have abundance of P cells or pacemaker cells, which is mainly responsible for generating its own rhythm, that means generating its own beat. So, this principle is known as atomicity. So, this are the two main differences between the myocardial cells. Myocardium, we have atrial and the ventricular myocardial fibers. Modified myocardium is present in the internodal pathways, the nodal tissues. We have bundle of His and the Purkinje fibers. So, first we will talk about the SA node or the sinoatrial node. Now, sinoatrial node is the primary or the main pacemaker tissue of heart. Why it is called the pacemaker tissue of heart? Because it contains abundant of P cells. Now, these P cells are present everywhere in the heart, but the proportion to which it is present, it varies. So, SA node or the sinoatrial node consists of the maximum number of P cells, and SA node, where it lies, SA node usually lies close to the opening of the superior vena cava. It usually lies in the right atrium, right side. So, right atrium at the opening of the superior vena cava. So, this sinoatrial node has got a diameter of 0.5 centimeter, and the action potentials from sinoatrial node are of slow type. Now, we also know the resting membrane potential. We had studied of different muscles are different. In cardiac muscles also, we have the cardiac myocytes of resting membrane potential, ventricular myocytes minus 90 millivolt, but nodal tissue, specifically SA node, consists of RMP of minus 50 millivolt. So, the action potentials in case of SA node is slow type, which is mainly because of the calcium ion influx, and the velocity of the conduction of this impulse is also slow, that is 0.05 meter per second. Now, this is the sinus node or the SA node, and this we have the AV node. Now, AV node is the atrioventricular node. This lies just above the atrioventricular ring, and the fiber diameter is also small in case of AV node, and the rate of impulse or the conduction is also slow. That means it is slower than that of the SA node also. Usually it ranges between 0.02 meter per second to 0.05 meter per second. So, this is the conduction speed of the AV node. Now, why the conduction at the AV node is slow when it is also one of the nodal tissue as just like that of the SA node. Now, if I draw the AV node simply, so this AV node consists of various cells. So, this AV node also consisting of various P cells. The number one, the first thing is in AV node the gap junctions. Gap junctions are the electrical window which connect the two cells. These are comparatively less. That is why the conduction is slow. The second thing is because of the fiber diameter. The nodal fiber diameter is small. Now, the smaller the diameter, the resistance to the flow of anything will be high. So, whether it is impulse or whether it is blood flow, whether it is air flow, it is always high the smaller the diameter of the lumen or the diameter of the fiber. The third thing is the resting membrane potential in case of AV node is minus 60 millivolt. So, normally we see the ventricular myocytes is having resting membrane potential of minus 90 millivolt. Now, depolarization means influx of positive ions or cations. So, the more the negative the charge it will be, the more negative the membrane potential it will be. So, there will be more attraction or more rushing of the positive ions towards it. So, since minus 60 millivolt is also negative potential, but minus 90 millivolt in the ventricular muscle, that is further more negative. So, resting membrane potential of AV node is comparatively less negative to the other ventricular myocytes. So, these are the very important features of AV node which makes the AV nodal conduction very slow, and there is a particular significance of this delay in the AV nodal conduction, and this AV nodal conduction, because of this slow conduction, that there is usually a delay of 100 milliseconds or 0.1 seconds, and this delay is very much significant because whenever there is a passage of impulse from SA node to AV node, the impulse is passing from SA node to AV node. So, whenever there is an impulse passage from SA node to AV node, depolarization is occurring. So, depolarization is the electrical event. The mechanical event that will occur after the depolarization is the contraction. So, AV nodal delay is very much required to ensure the atria are into maximum contracted state, so that the ventricles are fully filled. Otherwise, there will be half filling of the ventricles and there will be less stroke volume and so the cardiac output. So, to ensure the maximum contraction of the atria and proper filling of the ventricles, there is slow conduction or there is AV nodal delay of 100 milliseconds. Now, this again will come later. So, before that there are three nodal pathways between SA node to AV node. Initially, it was thought that the impulse from SA node to AV node traverses across the atrial myocytes, but later the ultrastructure has said there are three nodal pathways between SA node and the AV node. The first one is anterior internodal pathway, the middle internodal pathway and the posterior internodal pathway. So, this is anterior internodal pathway, this is middle internodal pathway and this is posterior internodal pathway. Now, anterior internodal pathway rises from the anterior surface of the SA node to the anterior surface of the AV node. Now, it also gives another branch to the left atrium, direct branch it gives to the left atrium. So, internodal pathway, anterior internodal pathway is giving two branches. The first branch is directly to the AV node and the another branch it directly gives to the left atrium. That branch is known as tract of Bachmann or Bachmann tract. Now, the middle internodal pathway, also known as tract of Wenck-Bach, generally supplies the interatrial septum, and the posterior internodal pathway is the tract of Thorel, which usually traverses from the posterior surface of the SA node to the posterior surface of the AV node. So, this is all about the internodal pathways between the SA node and AV node. So, the impulse traverses from SA node to AV node. This signifies nothing but atrial depolarization. So, when the impulse is traversing from SA node to AV node, this is atrial depolarization. That means now atria will contract. Only the atria will contract because atrial myocardium and the ventricular myocardium are separated by annulus fibrosus, except the AV bundle where already the depolarization is slow. So, initially the atria will contract. Now, coming to the rate of atomicity. This rate of atomicity is very important because SA node is the primary or the main pacemaker in our heart, but AV node and his bundle or Purkinje fibers, these are known as latent pacemaker. Now, why this is all known as latent pacemaker and why SA node is the main pacemaker? Because SA node conduction is the conduction in the SA node takes place in such a way because of the presence of many number of P cells, 60 to 80 beats per minute it generates. That is the rhythm of the heart. Generally, our average heart rate is 72 beats per minute, this we know. So, this is the average heart rate. So, SA node is the sinoatrial node which controls our beats, and it ranges between the 60 to, or it sends the impulses in between 60 to 80 beats per minute. Maximum it can go up to 100 beats per minute. Then coming to the AV node, it could beat AV node as well as bundle of Hess at the rate of 40 to 60 beats per minute. The range I am talking about 40 to 60 beats per minute, and Purkinje fibers they beat at 15 to 40 beats per minute. Now, this is because of the number of P cells which are present in these tissues. Now, SA node usually inhibits or keeps a check on the other nodal tissues, whether it is AV node or His bundle or Purkinje fibers. So, this is known as overdrive suppression. Why it is known as overdrive suppression? And because of this overdrive suppression, SA node is considered to be the main pacemaker of our body, because it inhibits the other tissues from generating its own rhythm, and it only allows its own rhythm to carry forward. So, because of the overdrive suppression. But suppose there is a heart block at the level of His bundle. Whenever there is a complete heart block at the level of His bundle. So, what will happen? The impulses are not getting transferred from SA node to AV node. So, what the ventricles initially in this person, what will happen? The ventricles will take its own rhythm. That means, the ventricles will generate its own rhythm, and the rhythm of the ventricles nothing but the Purkinje fibers actually, that is 15 to 40 beats per minute. So, SA node inhibition is usually removed in cases of heart block or abnormal situations. Like I told you, at the level of His bundle, whenever there is a complete heart block, at that time the ventricles will generate its own rhythm. So, when that rhythm which ventricle it will generate, that is at the rate of 15 to 40 beats per minute, that is known as idioventricular rhythm. So, normally the question is asked, what is the rate of this idioventricular rhythm, which we had already discussed, that is the Purkinje fibers, and 15 to 40 beats per minute is the rate of the idioventricular rhythm. So, this is the role of atomicity. Now, already I had discussed that there is a delay of 0.1 second which occurs in the AV node due to various reasons, and this is known as AV nodal delay. Now, the ability of this AV node to slow down the electrical impulses which are coming from the SA node to the ventricle is known as decremental conduction. Now, this decremental conduction is very important because suppose in case of abnormal situations, suppose atrial fibrillation, any sort of arrhythmia related to atria, where there is atrial fibrillation or atrial flutter, the atria, suppose it is beating at the rate of very fast heart rate, suppose 300 beats per minute. Now, if 300 beats per minute is the net heart rate of a person, you can imagine that person will immediately go into heart failure. There will be sudden cardiac arrest. So, what it does, our AV node is the gatekeeper at this time. It will slow down the electrical impulses, it will slow down the heart rate whichever it is getting from the atria, that is from 300 to approximately 100 to 150 beats per minute, and the person will generate its heart rate as 100 to 150 beats per minute. So, decremental conduction is very important in slowing down the transmission of very rapid impulses from SA node to ventricles. Next coming to the His bundle. So, His bundle, what happens? His bundle is also a connective tissue, and this septum rises from the AV node and terminates at the level of the Purkinje fibers or the apex of the ventricle. So, His bundle on entering the interventricular septum divides into usually right bundle branch and left bundle branch. Now, right bundle branch is longer and thinner because right ventricle is comparatively the thickness is less compared to the left ventricle. This is right ventricle and this is left ventricle, whereas the left ventricle, the left bundle branch, this usually gives two branches. The left bundle branch, this usually gives rise to two branches. One is anterior fascicle, the other one is posterior fascicle. It gives an anterior branch and a posterior branch. Anterior branch, now right bundle branch, as I told you, it usually supplies the right ventricle, whereas left bundle branch, it usually supplies the anterior fascicle, supplies the anterior part of the left ventricle, and the posterolateral portion of the left ventricle is supplied by the posterior branch of the left bundle branch. Now, this left bundle branch, while supplying to the ventricular walls, gives rise to various fibers. You can see over here, these are giving rises to various fibers. These fibers are nothing but the Purkinje fibers. These are very fast fibers, and this Purkinje fibers, these branches are given off at the subendocardial layers of the myocardium. Subendocardial means this is the wherever it is giving the branches, this is the inner side of the myocardium, and here we have the apex of the ventricle. So, this is the outer side of the myocardium. So, towards the inner side of the myocardium, the Purkinje cells or these fast conducting fibers are given branches. So, next we come to the Purkinje fibers. So, Purkinje cells are the largest cells in the heart. The Purkinje cells are usually discovered by the scientist Johann Purkinje, and this person has discovered Purkinje fibers in the heart as well as Purkinje cells in the cerebellum. So, this is a network of fast conducting fibers that is present in the subendocardial regions of the right and the left ventricle. Numerous gap junctions are present. This is one of the reasons for the fast conduction of the Purkinje fibers. Now, why the conduction is fast? The first point is gap junctions are present in large amount. The second point is the fast action type of potential, fast action potential, because the ventricular muscle has got a resting membrane potential of minus 90 millivolt. So, this action potential will be fast, which is usually carried out by the sodium influx. And the third point is the fiber, the diameter of the Purkinje fibers that is large. What we had seen in the AV node, it was small. So, since the diameter of the Purkinje fibers are large, so there will be less resistance to the flow, and so the rate of impulse conduction is quite high in the Purkinje fibers. The rate is usually 4 meter per second, and the action potentials which are generated over here is of fast type. So, we have seen various conducting fibers rate. 4 meter per second is very important, the maximum speed of conducting fibers, that is the Purkinje fibers in the heart, and very slow minimum we have at the AV node, that is 0.02 to 0.05, to be very specific, 0.02 meter per second. So, this is the conduction velocity. SA node 0.05 meter per second, internodal pathways 1, AV node, this you have to remember, 0.02 to 0.05, Purkinje fibers 4 and other 1 meter per second. So, even if you do not remember all, the maximum and the minimum is very important, that is Purkinje fibers consist of the maximum conduction velocity, while AV node consists of the minimum conduction velocity. Now, coming to the conducting system of heart, how the electric impulse gets conducted from SA node to the ventricle. So, that means we will see, we have seen the electrical events, that is the how action potential is generated. There is a depolarization phase, there is a repolarization phase. We will see that how it occurs in the conducting system, how that depolarization wave or repolarization wave is conducted throughout the heart. Now, the impulse generation, as already told, that is sinus node. So, sinus node has generated its impulse, and the impulse direction is towards AV node. Now, since sinus node is situated on the right atrium, so the impulse will be directed towards left side downward. So, the AV node is also downward. So, sinus node, the impulse coming from the sinus node or sinoatrial node is left side downwards. So, this is the impulse, this is nothing but the atrial depolarization, and as I told you that this AV node is very slow conducting pathway, and this AV node will have a delay of 100 milliseconds or 0.1 seconds delay, and the conduction velocity at this AV node is 0.02 meter per second, which is very slow, to ensure full contraction of the muscles and proper filling of the ventricles. So, this AV nodal delay has been told. Now, there is a range given in the Harrison of AV nodal delay, that is 60 to 120 milliseconds of the AV nodal delay. If a specific answer is not asked of 100 milliseconds or 0.1 milliseconds, the range is sometimes given or asked, that is 60 to 120 milliseconds. So, after this AV node, Purkinje fibers has got maximum conduction velocity. Now, after this AV, the passage of the impulse through the AV node or the AV bundle, then it will pass through the His or the bundle of His. Okay. So, this bundle of His, and at this level of bundle of His, the time it is taken at the bundle of His is 40 milliseconds. Now, before that I would tell what is the time taken for the impulse to travel from SA node to the tip of AV node, that is 30 milliseconds. So, 30 milliseconds SA node to AV node, then 100 milliseconds at the level of AV node, which has got an AV nodal delay, then we have 40 milliseconds at the level of His bundle. So, these are the time taken. If we start with 0 from here, so generally at the level of His bundle, it has taken around 130, 40 and 30, around 170 milliseconds. That means, the depolarization will start from the level of 170 milliseconds. So, from 170 milliseconds till the apex of the ventricles, the depolarization will reach at 200 milliseconds, which means now the depolarization occurs in various phases. The main 3 phases of depolarization which takes place, the first one is septal depolarization. Initially there will be septal depolarization, that means from left bundle branch towards the right bundle branch. So, from the left side to right side, so there will be septal depolarization. The next depolarization, that is the whole ventricular musculature, whole ventricular depolarization, and the last portion of the heart to get depolarized, that is the basal portion of the heart. The basal portion of the heart gets lastly depolarized. So, these three steps of depolarizations occurs one by one. So, after septal depolarization or the onset of septal depolarization start at the level of 170 milliseconds, that means when it reaches the His bundle, and at the apex, that means the ventricular depolarization takes place at the level of 200 milliseconds. Till now the whole ventricles have not got complete depolarization. It will only get completed when the basal portions of the heart also gets depolarized. So, the basal portion of the heart takes another 20 milliseconds, and hence the whole ventricle depolarization occurs at 220 milliseconds. These time intervals are very important because these are usually asked. So, the whole depolarization takes at 220 milliseconds, which means the Purkinje fibers takes only 220 minus 200 if we do 20 milliseconds to reach from this ventricular wall to the base of the heart. So, which means you can understand what could be the speed, how fast the conducting fibers are, the Purkinje fibers are. So, the apex of the ventricles 200 milliseconds. So, 200 milliseconds apex of the ventricles, and the whole ventricular depolarization takes place at 220 milliseconds. The rest part is the repolarizations. We already know the action potential, it ranges between 250 to 300 milliseconds in case of cardiac action potential. So, this is all about the conducting system of the heart, the seconds, the time intervals, which are also important. Now, the slower conduction velocity in AV node is already I had told you because of the small size and the slow upstroke of the action potential which occurs due to the slow voltage calcium channels, that means the slow action potential because of the resting membrane potential which is of minus 50 to minus 60 millivolt, and weak electrical coupling because of the presence of relatively few gap junctions. The importance of AV nodal delay allows proper ventricular filling, so that to ensure proper stroke volume and maximum cardiac output. It maintains low ventricular rate, that is the decremental conduction, to prevent higher impulses, higher rate impulses travelling from atria to ventricles. Also, there are effects of drugs which reduce the heart rate, partly by acting at the level of AV node by delaying the or promoting the AV nodal delay, that means increasing the AV nodal delay. So, these are the few examples, digitalis and digoxin or beta blockers. Now, digitalis and digoxin's mechanism of actions are already been discussed, that is, it is a blocker of the sodium potassium ATPase pump, and beta blockers, they act on the beta 1 receptors present in the heart. So, by blocking the beta 1 receptors, they will cause decrease in the cyclic AMP and hence in decrease in the calcium influx. So, these are the effects of the drugs. Also, vagal stimulation. Now, this AV nodal delay is subjective to change. Now, whenever there is a sympathetic stimulation, suppose there is an epinephrine secretions or fear, anxieties, whenever there is a sympathetic stimulation, this AV nodal delay will get shortened. Whenever there is a parasympathetic stimulation, that means vagal stimulation because of the acetylcholine. So, release of the acetylcholine. So, this AV nodal delay will get prolonged. So, this causes AV nodal block in subsequent constant vagal stimulation. So, coming to the conduction, the direction of the conductions in the heart. The first, as I already told you, the SA node to AV node, it is directed which from up towards the lower side, that means the direction is towards the lower side, laterals, lower and left side, and this usually contributes for the atrial depolarization. At the level of AV node, there is AV nodal delay of 100 milliseconds. Then moving to the His bundle, then at the level of the His bundle, it divides into right bundle branch and left bundle branch. At the bundle at the interventricular septum, there will be septal depolarization fast. So, septal depolarization fast. Now, the septal depolarization is from left to right side, because it has been said in some books that the septal the ventricular septum which is present, and here the left bundle branch, there is a connection with the left bundle branch. So, the left bundle branch is connected towards the it has got some connections with the septum, and so the depolarization takes place from left to right. After the septal depolarization, there will be depolarization of the ventricular muscle from the endocardium to the epicardium. Now, endocardium to the epicardium means from the inner side of the myocardium towards the outer side, from inner towards the outer side, because the Purkinje cells which are present over here, these are the maximum conducting fibers, and these conducting fibers are present at the level of subendocardial surface. Now, subendocardial surface means it is towards the inner myocardium, and so the depolarization wave will pass from the inner myocardium to the outer myocardium. So, inner myocardium towards the outer myocardium, if this is the apex, and so this is the inner myocardium and this is the outer myocardium or epicardium. So, the depolarization will be from inner side towards the outer side, that means from endocardium to the epicardium, and the last portion to gets depolarized, that is the basal portion of the heart. So, posterobasal portion of the hearts are the last portion to gets depolarized. So, this the start is the septum, that also which part of the septum? Middle part of the septum, left side is left and the portion is middle. So, left middle portion of the septum will get depolarized first. Direction is left to right, and the last portion of the heart to get depolarized, that is the base of the heart, that is left posterobasal region of the heart, and the depolarization, ventricular depolarization is from endocardium to epicardium. The opposite occurs in case of repolarization. Remember, the first side, the last side to get depolarized is the first side to get repolarized. Why it is so? Because whenever there is a contraction, depolarization means the contraction will occur, the muscles contraction will occur. So, whenever ventricles will contract to its maximum, so what will happen? The inner portion of the blood vessels will get constricted, there will be restriction to the flow of the blood, but the outer blood vessels, the outer myocardial blood vessels will remain sort of unaffected. So, the repolarization starts at the epicardial layer or the outer myocardium. So, the repolarization at the apex starts from the epicardium to endocardium. So, that is why this is the repolarization which starts from the epicardium to endocardium. So, this we have to remember. The action potentials from SN node are of slow type, and in case of Purkinje fibers, fast type. There is a AV nodal delay of 0.1 second, which is very important for the proper ventricular filling, ensuring proper cardiac output. Depolarization occurs always from inner to outward, that is endocardium to epicardium, while repolarization occurs from outer to inner, that is from epicardium to endocardium. So, these are the concluding topics, and these are the references. So, we conclude today's lecture. Thank you.